Hello all, welcome to my presentation on molecular dynamics and protein-protein interactions, in short PPIs on SARS-CoV-2 coronavirus. And I'm honored to have this opportunity to share my research with you today. I must thank my advisor Dr. Chloe Ixinxia for her constant support and guidance. These are the different sections that we will be discussing today. Coming to the introduction, this section provides an introduction to two key concepts that underline our research, which are molecular dynamic simulation and analysis of hydrogen bonds and salt bridges. Coming to molecular dynamic simulation, it is a powerful computational tool that enables the study of atomic level interactions between biological molecules such as proteins, which provides insights into the structures and dynamics. The analysis of hydrogen bond and salt bridges provide critical information about stability, dynamics, and specificity of these interactions, which is really essential for understanding various biological processes. Our research focuses on interaction of six variants of COVID with HACE2 protein. Here are the six variants. The orange color structure represents the original, silver color represents alpha, green color represents beta, yellow color represents gamma, purple color represents delta, and pink color represents omicron. Here are the tools and methods that we have followed. We have used camera to generate the structures of SARS-CoV-2 variants and along with interactive part of HAC2 protein. HAC2 protein is a protein in humans which is responsible for the interaction of COVID with human. And we have used camera to align all these structures to remove non-similar regions, and molecular dynamic simulation were run using NAMD2 tool, which is aided by a protein structure file generated by VMD software. A 10 nanosecond simulation was conducted to study the protein-protein interaction and assess its stability. But we have considered the results of MD simulations from 5 nanoseconds to 10 nanoseconds only because in the first 5 nanoseconds all the variants are highly unstable. Hydrogen bonds and salvages data were generated using VMD from the results of MD simulations. With Python, we converted this data into CSV format for easy analysis. Microsoft Excel was then used to generate these visualizations into interactions to understand its strength and stability. And here are the results of our research, and this is the for original variant. We have bar chart representing an hydrogen bonds in all the variants, and here are the hydrogen bonds with their respective occupancies. Original has a hydrogen bond with highest occupancy of 95.8%, and it has three salt bridges with an standard line at distance 3.2 angstroms, which is used to evaluate the strength of a salt bridge. If the distance of a salt bridge is consistently below the standard line, then it is a strong salt bridge. If the distance is consistently above the standard line, then it is a weak salt bridge. If the distance is moving around the standard line, then it is an mid-ranged salt bridge. The original variant has one strong salt bridge and two mid-range salt bridge. And coming to alpha, it has hydrogen bond of highest occupancy 94.91% and two strong salt bridges. And coming to beta, it has hydrogen bond of highest occupancy 95.5% and two weak salt bridge and one mid-range salt bridge and one strong salt bridge. Coming to delta, it has highest occupancy of 96.4% and it has two strong salt bridges and one weak salt bridge. And coming to gamma, it has hydrogen bond of highest occupancy 97.2% and it has only one salt bridge which is strong. And coming to omicron, it has hydrogen bond of highest occupancy 92.91% and it has four salt bridges. Among those, two are weak and two are mid-ranged. And coming to conclusion of our study has provided valuable insights into molecular behavior and protein-protein interactions of COVID variants with HACE2. Our findings suggest that hydrogen bond and salt bridge interactions play a critical role in stabilizing protein-protein interactions across all the variants. We have identified key amino acids involved in forming these interactions such as arginine and aspartic acid. Furthermore, we have identified differences in among the variants, which results gamma variant exhibited the highest hydrogen bond occupancy, while alpha and delta variant had the highest number of salt bridges with shorter distances. Notably, the delta variant formed the most stable interaction with HAC2 among all the variants analyzed. We hope that our findings will contribute the global effort to combat the COVID-19 pandemic. Thank you. Here is my contact email. You can reach out to me at any time. Thank you.